Okay, so we've talked a lot about cells, and we've talked about chemistry, of course. And when we talked about cells, we talked about some of the organelles. So we had the cell, and then we had the nucleus, and then we had things like the ER and the Golgi. And then we talked about secretion of certain things, okay? So, and we kind of stopped there. So now it's time to talk about what these things might be doing, okay? So we know that cells usually are in groups, whether it's epithelial cells or a lot of times like in your lymph nodes, you have different immune cells. And these guys have to communicate with each other so everybody knows what's going on, okay? So we have to know about this on some level because that's where drugs have their effect. So if you're talking about like a neuron, the neuron will communicate with another neuron, and there are chemicals that are released, neurotransmitters. And so there have to be some kind of a receptor here, so this one can communicate with this one, okay? And so that happens in your liver, it happens all over the place. Every single cell is somehow communicating with the other cells around it. So we're going to talk about some of those things, okay? So when we talk about, I guess, the reasons and the mechanisms for how these cells communicate, remember we've mentioned that the inside of the cell is about minus 70 millivolts, okay? If that changes, if that goes up or if that goes down, that cell has some kind of an effect, okay? So that cell's going to have some kind of a response. That means something to the cell. So, and we call that a change in the membrane potential, okay? So this area across here, this is a potential, okay? So that's the ability for charges to move. So ions might move in here, and that might make it minus 60, okay? And so that's how neurons know what to do. That's how they communicate. That's how they function. That's how muscle cells function, and that's how other cells functions, including things like the egg during fertilization. There's a change in the potential. Okay, and then there are also chemical signals. Okay, so like the neuron. Okay, so there's an electrical signal that's happening down this axon and up here in the cell body. But then there are neurotransmitters that are released, and that's going to be a chemical signal. Okay, so that's released, secreted by cells into the extracellular fluid. Okay, so that's what's happening there. And then, of course, as I mentioned, immune cells, they have to communicate with other immune cells, and so you have a response. Okay. And so they're going to change some of their intracellular functions so they do something different, like attack like a bacteria or a virus or something and wipe it out. All right, so chemical signals are responsible for most communication. Now we're going to talk about the electrical signaling when we cover neuro part of things, and when we talk about muscles, and when we talk about heart even. And so there have to be target cells. So if this cell releases a signal, it has to have a target. When this neurotransmitter releases its signal, it has to have a target. Okay, so this is its target. And it may branch out, it may have a whole bunch of targets. So there has to be a target cell and something to receive that signal. So we have to discuss some terms. Okay, so here you can see this cell down here, this guy right here. And somehow cell signaling always, I mean, there's a communication coming in and there's a communication leaving. So somehow this guy got a signal or something that said, okay, I need to release a signaling molecule. Okay, so this is what this is right here. This is a signaling molecule. Now, if you look at this signaling molecule can actually bind to the same cell that just released the signaling molecule. And this happens. It's called autocrine signaling. Auto means self. Okay, so it signals itself. Now, or it may, and usually it does both, it may signal other cells. So this cell over here is receiving the signal, and then this cell is receiving the signal. Now, if it's acting on another cell in the area, so or adjacent cells, some kind of cell in the area, we call that paracrine signaling. So the only difference between autocrine and paracrine is actually being released by the cell and then it affects that cell. Paracrine, it's released by a cell and it's affecting other cells in the area. Okay, so they have to be close, or what we would say is local cell-to-cell communication. And I'll just say very quickly, a lot of people wonder why a cell would release a molecule and then bind to itself. Probably because this cell has two different communication types of functions. Okay, so it gets a signal in and then it causes it to release a molecule. Okay, now it's releasing this molecule so it can tell these cells, hey, guys, something happened to me and I need to tell you about it so you can have some kind of a response. Okay, okay. So if this one also, it's the same kind of cell, so it also has to have a response. Now, rather than having a separate mechanism in here which tells it to respond, it uses the same method as everything else, and it has this method that then tells it to respond. So if that makes any sense, you have a signal coming in saying, hey, something's going on, and it has two things that it can do. It can send out a signal, and once it receives a signal, it can respond. Okay? And that's all any of these cells need. All this one needs is, oh, I got a signal, I can respond. The only other job I can do is release a signal. Okay, so two things. This one, if it were going to have a different mechanism, it would have to say, oh, I received a signal. Now I'm going to have a completely different mechanism so I can respond, or I can just use this thing I already have. I already have a receptor up here that causes me to respond, so I'll just use that. And so it's a much simpler way to have an autocrine signal. And so that does pose a lot of questions for people why autocrine signals happen, and it's because you don't have to have a third intracellular mechanism. You can just use this one up here. Okay, so I hope that makes sense. So those are autocrine and paracrine, and those are local. Now, what if we have to communicate with something a long ways away? Okay, so what if we have to change our digestion and we want to release a signal, say, from the pituitary gland or from the thyroid gland or something like that? And that happens through, and this is the important thing here, the blood. Okay, so think about that. The blood goes everywhere in the body. So all you need to do is you need to have an area, we call it an endocrine cell or an endocrine gland, like the thyroid gland, and the pancreas has some endocrine function. It releases insulin and glucagon. And so we can have this sitting here in a localized area a lot of times, normally, and it will release a molecule, and that molecule can go all over the body. Okay, so the thyroid gland affects lots of different areas, but it's just located in one little area. This is supposed to be a person. It's just located in one little area in the neck area. Okay, so that's a guy, and that's his neck. Okay, and that's the thyroid gland. So that can be released, and then it can have its effect in lots of places. Okay, all right. So the question is, if you're releasing it into the blood, it's going to affect every cell, right? Well, the answer is no, because you have to have some kind of a receptor. Now, this cell, until I drew it, doesn't have a receptor, so it's not going to be affected. This cell over here has a receptor, and so it's going to have a response. And that's very important because that's where the control comes from. Okay, so that's why you can have the thyroid gland just sitting up here in your neck, and you can affect cells anywhere else in the body. Okay, and it's because that's where the receptors are. Okay, so the important thing here, the thing I really want you to understand, is that when you're talking about hormones, you're talking about long distance signaling, and hormones are, by definition, released into the blood. Okay, so they're not just released into the extracellular fluid around them, they're released into the blood. They have to make their way into the blood so they can go everywhere. Okay, now, we have another set of hormones, and you'll hear this sometimes, neurohormones, things like epinephrine. They release into the blood just like a hormone, so they're still a hormone. They're still releasing it into the bloodstream. However, in this case, they're releasing instead of from an endocrine cell or an endocrine gland, which are made up of endocrine cells, it's releasing it from a neuron. So we give it a different name. We call it a neurohormone. okay? And then because it's released from a neuron. So hopefully that makes sense, and it's the same deal. If there's a receptor, it has an effect. If there is a receptor, like in this case, it has an effect, and if there is no receptor, it doesn't matter. This one just doesn't do anything. It doesn't change anything it's been doing. Okay, so now we've talked a little bit about neurons, and we've said that the neuron can release a molecule into the blood, and that makes it a neurohormone. Well, if it's released from a neuron into the extracellular fluid and not into the blood, and it just has its effect on the next neuron or cell, okay? So usually, and I say that because of sheer numbers, usually a neuron communicates with another neuron, but it doesn't have to. A neuron can sometimes release a neurotransmitter, okay? So that makes sense, neuro, and it's transmitting. So a neurotransmitter directly onto any kind of cell, okay? Now we have so many neurons that it's usually neuron to neuron, but this can take place, okay? And so they have a very rapid effect. It happens right away. And then, like I said, they have an electrical signal, and we call this an action potential. We'll talk more about that when we talk about the neuro section. So it has an action potential that runs down the neuron, and that's electrical, and then, or down the axon, and then the neurotransmitter is released and it has its effect. Okay, so it's very, very fast. It's very immediate. That's why we're able to think. Okay, so we've covered autocrine, paracrine, and then hormones, neurohormones, and neurotransmitters. Okay, so we have other types of cell-to-cell communication. And this one is very, very important for function. I mean, of course, everything is important. You can't survive without neurons. But these are called cytokines. Cytokines were actually first found in the immune system. Okay, so that's where we know them. Okay, so that's how we discovered them. I wasn't there, but that's how they were discovered. Is in the immune system, and they were trying to figure out how all of these cells, and you don't have to know all of these pathways, but it shows you how complicated it can be. They were wondering how all these immune cells knew, so when one cell was affected, when a bacteria or a virus invaded, it would have a response and it would tell all of the other cells to respond. Okay, so and then you had this huge immune response, and that's why we have swelling of our lymph nodes, because that's where a lot of these cells kind of rest, and that's where they stay most of the time. And so they would start to divide, and they would have this huge effect. So then we knew there was some kind of signaling going on, and so it was a process of trying to figure out what these signals were. Okay, and we finally found them. They finally found them, and they called them cytokines. Okay, and then they figured out that, and this is important, that all nucleated cells actually produce these cytokines. Okay, and now they may affect the cells in the area, which is what this kind of shows, or they may have long distance signaling. So they may affect cells in lots of different places of the body. So we can't really call them autocrine, paracrine, which is close or local, and we can't really call them hormones. And so they have their own little category. And they have their own names. They're usually called like IL, which stands for interleukin, which means between white blood cells, and some are just called, cytokines. Okay, and there are a lot of diseases associated with, like, for instance, this one. I don't know if you can read this, but it says, wait, not that one. If you have, there's one called IL-2, interleukin-2, and I don't see any on here. Which is very important for an immune response. Okay, I thought that we had one on there, but apparently we don't. So you can see here that there's a microbial infection or yeast infection or bacteria or a virus here. So bacteria, yeast, virus, and you can see that this is affected, and it will release this interleukin or this cytokine. Okay, and then it will have an inflammatory response. Okay, all right. So if we look at just in general how these things have their effects, we have to have a cell that releases whatever it's releasing, whether it's a cytokine or a neurotransmitter or something. It has to have a way to affect the next cell. Okay, so now sometimes if it's a hydrophilic, which is another, which they're calling it lipophobic here, but that's also hydrophilic, okay, signal, then it has to bind to a receptor. Has to bind to a receptor. Now, and so when it binds to this receptor, then it'll have its effect inside the cell. Now, this is going to be on quizzes, tests, something, and it always confuses people. Okay, so hopefully we understand this difference between hydrophobic and hydrophilic. Okay, hydrophilic. Remember, hydrophilic means that it likes water. It's attracted to water because of the electrical properties. Hydrophobic means that it doesn't really care about water. Okay, so this is hydrophobic. In this case, they're calling it lipophilic, which means that it likes lipids. Okay, so it is soluble in lipids. And what that means for us is that this hydrophobic / also lipophilic signal will go right through the membrane, which is very important because things like estrogens, a lot of the steroid molecules, they just are released into the blood and they'll just go all the way through cells. It doesn't really matter. So if that's the case, then you can have a receptor inside the cell. You can even have a receptor inside the nucleus. And so this molecule right here, because a lot of steroids actually turn on or off DNA. So they just have their signal or their receptor right in here, and you don't have to mess with anything else. Okay, so only lipophilic molecules can do that, only if they can move through this cell membrane. Now, if it's hydrophilic, now lipophilic, hydrophobic, are also listed here. Okay, so I circled it up here and I said, look, they can go all the way through. This is the part that confuses people though, is that here you can also see that sometimes they have a receptor that's out here on the membrane. It's as though they can't get through. Now this is probably because they don't go through as well. Okay, so you know that they can get the signal if they just bind to a receptor on the outside of the membrane. So I hope that makes sense. So that means that lipophilic, which I labeled here, and lipophilic, which I labeled here, they can either go through the membrane or they can bind to a receptor on the outside. They can do any of them. Now, if we look at the other ones, if we look at the hydrophilic, which in this case, they're talking about lipo because they want to make it clear that it's the lipid, that it either likes the lipid or it doesn't like the lipid. So hydrophilic, which is the same as lipophobic. I didn't invent all these terms. I just try to help you keep them straight, and I try to keep them straight myself. If a lipophobic, that means that it can't. That means that if this thing were to try to go through here, it wouldn't. It would just bounce right off. Okay? It's not able to penetrate the membrane. It can only bind to a receptor on the outside of the cell. So if it's lipid soluble, lipophobic, it can do any of these. It can go into the cytosol, it can go into the nucleus, or it can bind to a receptor. If it's hydrophilic, or what we would say lipophobic, it has to bind to a receptor on the outside of the cell. And then this will communicate with the inside of the cell. So that's what we're going to talk about next. Okay, so if you look at the different kinds of receptors there are, because remember, our goal is to have some kind of an effect inside the cell. So we want to have an effect inside the cell. So now we have these channels where, if you look at this little molecule here, this is extracellular. So it could have been released from anywhere, autocrine, paracrine, hormonal, whatever, neurotransmitter. So here's the molecule, and it can bind to a little receptor. And when it binds the receptor, it can have an effect. Okay, so here's another one. In this case, this is an enzyme. So you have a molecule from outside the cell activating an enzyme inside the cell, which is going to have an effect. Okay, this is another one, and this is what I'm going to talk about in a bit. It's called a G protein coupled receptor. And this can have, your binding happens here, and it will activate this, which has an effect inside the cell. This, I believe, is a tyrosine kinase, and which is a different kind of molecule, but it's the same thing. So it has, this is an integrin. And so it can have an effect on the inside of the cell or the cell structure. Okay. Now, once we get the signal into the cell, so if we look at this, so it converts chemical signals, so from outside, okay, into a cellular response, which is inside. Okay, so here's our signaling molecule. If we look up here, here's our signaling molecule. It binds to a receptor, and then it can have a whole lot of responses inside the cell. Okay. Lots of things can happen. It can actually cause a cell response directly, which is what we see here, or it can actually open up another channel. See this guy? There's no receptor up here for it, or there might be. It might need to do both. So, or this may not have any receptor at all, and by activating this one over here, you might activate this, and this will then open that channel. So somehow we're having a signal, and that's the big deal. We're having a signal from the outside that's having an effect on the inside, which is causing the cell to respond. So I want to say a little more about this G protein coupled receptor because there are questions on it on the exam and the quizzes about this. It's 7 transmembrane. So what that means, it's a protein. This is a protein itself, and the way that it's structured, it crosses the membrane 1, 2, 3, 4, 5, 6, 7 times, and then there's a little tail that's inside. So the receptor will bind up here, and it'll cause some kind of a shape change, what we call a conformational change, that will activate a G protein. Okay, so G protein is just a GTP instead of an ATP. Okay, so we're not too concerned about that, but it hydrolyzes GTP. So we're not too concerned about this, but that's what it is, and that's why it's called this, because we call this a G protein. It has a few different subunits, but then this will go on to cause something else to happen. Okay, so there's a cytoplasmic tail of the GPCR, G protein coupled receptor, that will then affect what we call a transducer or a signal communicator molecule. So this will communicate the signal to the rest of the cell. Okay, so they can do lots of things, open ion channels in the membrane, alter enzyme activity on the cytoplasmic side of the membrane, but they're doing something to the cell. Now the reason that I talk so much, or I mention G protein coupled receptors, GPCRs, is because about half of drugs work on these. Okay, so we're going to see things like agonists and antagonists. And so you could take a drug that may activate this or antagonize it, or you could take a drug that blocks it. Okay, so if it activates, I may have said that wrong. I don't remember, but if it activates it, it's an agonist. I think I may have said that backwards. And if it just kind of gets in the way, so it just kind of blocks it, that's an antagonist. Okay, and like I said, about half the drugs will have an effect. Half the drugs that we take have this effect because these are everywhere and they do a lot of different things. But in general, we say that they activate the inside of the cell. They do something inside the cell. Okay, so here's some examples. And this is, again, why they're so important. Again, we're still talking about G protein coupled receptors. And so you can see here that there's a signal molecule. One signal molecule. Now, if you're looking ahead of me, you can see that that one signal molecule has had a large effect. Okay, so it's activated. It's G protein. That's why it's called a G protein coupled receptor, because this is the G protein which activates another enzyme. This enzyme is called adenylyl cyclase, which turns ATP into something called cyclic AMP, which activates a kinase. Kinases put phosphate groups on things, so we say it's phosphorylated a different protein, which then goes on to activate something else, which then goes on to activate something else. Okay, so you don't have to know all of these mechanisms, but you do have to know that that's what a GPCR does. Okay, so it does. It can cause a huge effect on the inside of the cell, even though it's just one molecule. This one little molecule binds and activates all of this. Okay, so that's one way that they work. There are only a few different ways that they work, so it's not impossible to learn. But I think usually you learn that in a regular biology class or a cell biology class or something like that. I just want to either remind you about them or make you aware that these things exist, and to get that GPCR, G protein coupled receptor name into your head, the 7-transmembrane tied to a G protein. Okay, so here's a different mechanism that they use. So in this case, it's activating something called phospholipase C and protein kinase C, IP3, which is called inositol triphosphate. Lots of big names. You don't have to know all these. I'm not going to ask you about these. But you can see in this case, this is actually the goal of this when this guy binds is to release calcium. Okay, calcium is a signaling molecule. Okay. Okay, so calcium is a signaling molecule, and it's going to have lots of effects. Okay, so this just says cellular response, which works for us. And so together there's also phosphorylation that takes place. So lots of different things. And I don't mean to confuse you by it, but I just want you to see that there's some range to what GPCRs do, but ultimately they cause big changes inside. Okay. Okay. I don't know if that's a formal term, but that's what they do. They tend to change the inside of the cell. Okay. Sometimes they can be very quick, like in this case. They have their effect fairly quickly. So here's this is what I said, this is a channel molecule, and so even though this binds, it may not activate. Okay, it may be waiting for a signal from the inside, so you have to have something else that binds to the GPCR, which causes a cascade of responses, but one of those things is to go up here and open that, okay? And then it'll let ions in or out, okay? And so that changes the membrane potential. Now this would happen very quickly, okay? So it binds and then it immediately opens that signal. And a lot of things, like I said, these are everywhere. If you've heard of dopamine or serotonin, a lot of dopamine and serotonin neurotransmitters, that's what those are, will have their effect this way, okay? They may not even bind, there may not even be any binding to the receptor, it may to the channel. It may bind to the G protein coupled receptor, cause a whole bunch of stuff to be released, and then keep that channel open for quite a while. Okay, so there are lots of different ways, lots of variability and diversity in the way these guys work. Okay, all right. Now, some other signaling molecules. And I try to only talk about things that are somehow relevant, even if you never see it again, or if you've it doesn't even matter to you. But this is relevant. Nitric oxide, and I'll start here saying it is one of the most powerful vasodilators known. Okay, so vasodilation means that it takes a blood vessel that might be open this much, and it opens it this much. Okay, so there's blood flowing through here. Okay, and this will allow more blood to flow through. Okay, so vasodilation allows more blood to flow through the blood vessel. Okay, so if that's the blood vessel. All right, so it's important because there are heart medications that may do this. You may have heard of nitroglycerin. Nitroglycerin, you know, the little pill that some people put under their tongue when they feel like they're having angina or something like that, and that will dilate the blood vessels, which will lower the blood pressure. Okay, so it's like if you're trying to put a bunch of water through a hose and then you make that hose larger, it relieves the pressure. Okay, so that's what this does. That's what vasodilation does. Decreases the blood pressure, and that's really important. And it increases blood flow. Now, another place that's kind of interesting where this is well known is there's a little drug called Viagra, which started out as a blood pressure medication because ultimately it led to increases in nitric oxide. Okay, so it led to increases in nitric oxide and it vasodilated. The trouble is Viagra tended to vasodilate um, more in a specific region, and that would cause an erection. And so the drug company who was researching this thought, well, that seems useful, and so they made a lot of money by making Viagra. And all it does is it increases nitric oxide and it causes vasodilation. Okay. All right. Now, um, here's something else that will be asked about on exams, and I'll tell you why it's important. This is your lipid bilayer here. Okay, so those go down, and then you've got those things all the way down. This is a phospholipid. Now, what happens in this case is that we want to have a signal that occurs inside the cell, or we may want to release something out of the cell, but we have to make it first. Okay, so what happens sometimes is we'll actually take one of the tails of this phospholipid, change it around a little bit through this PLA2, phospholipase A2, and this is the enzyme that does it, and turn it into something called arachidonic acid. Okay, arachidonic acid sounds like spider. Okay, and that's because that's where they first discovered this. Was they discovered it in spiders, and this too is very important. This arachidonic acid, because it goes on to form something called prostaglandins, thromboxanes, and something called leukotrienes. Now, you may never have heard of any of those. I don't know. But prostaglandins cause pain, fever, inflammation. Okay, certain prostaglandins do. And so it would be handy if someone was suffering from pain, fever, and inflammation to block this. Okay, so this arachidonic acid, not let it turn into the prostaglandin. And so here's the enzyme that does that. It's called cyclooxygenase, COX-1 and COX-2. So wouldn't it be nice if we had a medication that would block that so we would not produce as much or as many of these prostaglandins? And we do. The next slide shows this as well, and we call those NSAIDs, non-steroidal anti-inflammatory drugs. Okay, so that's how ibuprofen and aspirin, that's how they work. They block this mechanism so the prostaglandins can't be made, and it reduces pain, fever, and inflammation. Okay, so that's pretty handy, I would say. Another thing, thromboxanes, this has to do with blood pressure, blood clotting, those kinds of things. And so we also see some effects with that. It thins the blood a little bit when you take aspirin, so we can take aspirin. So this molecule is important. And remember, it all just comes from phospholipids. Phospholipids turn into this molecule called arachidonic acid, and then it makes all of these things. Now there's this other thing over here called leukotrienes, and that's how you're supposed to say it. It's tri for 3 enes. Enes are double bonded molecules, so leukotrienes. And these guys show up in the lungs. Okay, so they might show up in other places too, but we know about them most because they show up in the lungs, and they are affected, or they play a role in asthma. Okay, so we're discovering more and more about this. A lot of research being done on these. So we're trying to find things that will specifically block these leukotrienes, especially the ones that are involved in asthma and use that as a very effective asthma treatment. Okay, so there's a lot of research being done on that. So you can see this is just we call that novel or different or new or something, signaling molecules that deal with these lipids, and they're phospholipids. Okay, and they go on to make all of these things. Now we've known about this, the cyclooxygenase, the COX blockers, that are like the ibuprofen and the other NSAIDs that block prostaglandins. But this is kind of a new thing. And then we've discovered the effects of aspirin on blood thinning and reducing heart attacks. Okay, so just to go over this real quickly, since I already kind of went through it, leukotrienes have a role in asthma and anaphylaxis. So a large immune response. That's what anaphylaxis is in general. And then the prostanoids, which are the prostaglandins that I was talking about, inflammation, pain, fever, sleep. I didn't mention that. Thromboxanes, which have their effects on, they basically thin the blood and they prevent clots. Okay, so that's what aspirin does. The thromboxanes will promote that. So if we block those, then we reduce our chances of a heart attack or a stroke. Okay. And that's the other thing I mentioned. Non-steroidal anti-inflammatory drugs help prevent the inflammation by inhibiting this cyclooxygenase or COX. And that's what, again, I've already shown this, but that's what we see here in NSAIDs. They block cyclooxygenase. So this arachidonic acid can't turn into these things. Okay. All right. So hopefully that makes sense. All right. So now modulation of signaling pathways. So we care about specificity, competition, and so what are we talking about? We're talking about a cell being here, and there's a receptor. Okay, so here's a receptor, and the signaling molecule is supposed to come down and bind there, but like I said, we can put something there and block it, or we can activate it directly. Okay, and so it's a competitive binding. So we put something there that's going to compete with this, and it might lower the function, or we put something there that actually activates it, and we're going to increase function. Okay, so we have a little bit of control over that, and that's what drugs tend to do. Okay, so what I'm talking about, again, I mentioned this before, is an agonist versus an antagonist. Okay. So an agonist is a drug, usually, that activates it, and an antagonist is a drug, usually, that gets in the way. It blocks it. It doesn't turn it off, necessarily, but it gets in the way so this guy can't bind it. Okay. So sometimes, like in the case with epinephrine, or epinephrine, another word for epinephrine is adrenaline, I could ask the question, does adrenaline cause vasoconstriction, or does it cause vasodilation? So does it cause your blood vessels to get smaller? So if this is the original size, does it cause them to get smaller, or does it cause them to get bigger? Okay, and the answer is both. Okay, it can actually do both, because there could be different, that's what this says right here, multiple receptors for one ligand. So here's adrenaline, epinephrine, same thing. We call it epinephrine. England calls it adrenaline, and we also call it adrenaline, depending on our usage. But in one case, this epinephrine might cause vasoconstriction, might cause it to get smaller. I just turned this from a cell into a blood vessel. So now this is a blood vessel. Sorry about that. So epinephrine could bind and cause this to constrict, or vasopressin could bind, or I'm sorry, not vasopressin, epinephrine could bind and cause it to dilate, okay, get larger. Okay, so remember, it's the same drug, it just has different receptors for that same drug, okay, or for that same, in this case, it's a hormone, okay, neurohormone. All right, so, and we can also, as we know, we can also give this as a drug. We can give adrenaline or epinephrine. We can administer that. Okay, so this is a better picture, hopefully, to two things. One, to show you a little more clearly, but the other is to make sure that you know that it's important. An agonist versus an antagonist. Okay, so picture one. The primary ligand activates the receptor. So if we talk about something like epinephrine or dopamine or something like that, that's the primary ligand. That's what we get. That's what we make in our body. So this is the body functioning normally. The ligand binds the receptor. That's the definition of a ligand, something that binds the receptor. And so it binds the receptor and it causes a response. Okay. Now, an agonist, we could give a drug that's not the same thing, or sometimes we give drugs that are the same thing, but we can give a drug that's different than the regular ligand that we produce, but it still activates it. Okay, so that is an agonist. Okay, it agonizes or it makes it happen. Okay, and it has a response. It gives a response. Now, this is showing the normal ligand. So this is what the ligand would normally do here on the left again, and it would have a response. Now, we might take a drug, a lot of schizophrenia drugs are like this, that will come down and bind and do nothing. Okay? Now, that's where a lot of people miss the questions on this, is when you try to describe what an antagonist is and you want to say that it has the opposite effect. It doesn't have the opposite effect. It has a nothing effect. It simply gets in the way. So the body is saying, "Ooh, gosh, we need to activate this receptor. We need to cause a response," but there's an antagonist sitting on it. We've taken a drug, some kind of a medication that's sitting right here. And so when this guy over here tries to bind, it's rejected. Okay, it can't bind. So the agonist activates, the antagonist blocks the activation. Okay, so it doesn't let it react, or it doesn't let it have a response. So there's no response. Okay, not an opposite response, just no response at all. Okay. So this is a picture in case you missed it earlier. So this is if we were to administer epinephrine or adrenaline. We're in America, so we should call this epinephrine, or at least I'm in America. So this is an alpha. We would call it an alpha adrenergic receptor. This is a beta adrenergic receptor. Same ligand. Same thing can bind to both of these, and it does. It binds to both of these. Okay, so I asked earlier, what happens when you release adrenaline? Do you have vasodilation, which we see here, or do you have vasoconstriction? I said the answer is both. But it depends on where it is, right? Because if you have an adrenaline rush, you definitely want to increase blood flow to your skeletal muscles, okay? Because you want to be able to run away if you have to. But you don't necessarily, so that would be what you would see here to the skeletal muscles, but you don't necessarily want to increase blood flow to your digestion. Okay, so that's not the time to be digesting the cheeseburger you ate. So you reduce blood flow to those areas. And that's, if you think about that, that's pretty powerful. You have an epinephrine or an adrenaline release, and your body, all you have to do is you have one ligand, epinephrine, and all you have to do is put different receptors for the same ligand in different places. One of them causes vasodilation, one of them causes vasoconstriction. Okay, so hopefully we went through that in good enough detail that you understand it. So there we go.